When a brain gets old, we picture neurons dying. In naturally aged mice, the sharpest damage sits somewhere else. It's in the wiring, not the thinking cells. And it traces to the brain's own cleanup crew, breaking down in one exact spot. The researchers stained aged mouse brains for a senescence marker, galectin-3. In young brains, it was rare. In aged white matter, it was dense, thickest in one tract, the fimbria. The marker sat on microglia, the brain's cleanup cells, not on the astrocytes. So this is one cell type, in one place. When we picture an aging brain, we picture neurons dying in the hippocampus. But here, the senescence and damage genes ran higher in the wiring than in the hippocampus, in both males and females. The signal was in the cables, not the processors. A separate human study saw the same shape. It followed 102 older adults with healthy memory. The ones who lost a wiring tract, the fornix, were the ones who declined. Their grey matter gave no warning. So why would a cleanup cell break? Here's the model the authors propose. Microglia clear worn-out insulation from the brain's wiring, and that job never stops. Swallow enough debris, and the cell's disposal bags overload. Overloaded, they spring leaks. Galectin-3 rushes in and piles up at the leaks. That's why the marker sits on these cells at all. Then the cell locks into a senescent inflamed state it cannot leave. Senescent doesn't mean dead. It means locked on. Galectin-3 is the fingerprint of a broken garbage disposal. But why this tract, and not the one beside it? The authors point to geography. The fimbria sits right against the choroid plexus and the fluid-filled ventricle. That leaves it unusually exposed to whatever the blood and brain fluid carry. And they posit that this one spot explains two opposite things: why the fimbria breaks first, and why it responds first. Single-cell imaging found a group of microglia that appears only in old brains, carrying both the damage and senescence marks. Can any of it be undone? In aged female mice, the team cleared the p16 cells. In the fimbria, three things reset. The microglia thinned back to youthful numbers. The swollen cells shrank. And the cells that had lined up along the nerve fibres swung back toward youth. 5 or 6 mice per group. The authors call it partial. And nobody tested whether the mice could think any better. But here's the catch. The tool that did the most is not a drug. It's a genetic self-destruct switch that only works in an engineered mouse. The one that is a drug, venetoclax, approved for leukemia, cut a damage marker, but never reduced p16. And it altered young brains too. Here's what nobody has settled. It isn't clear that these treatments acted on the brain at all. The authors say so plainly. It's unclear whether the drugs reach the brain and act there directly, or whether they clear senescent cells elsewhere in the body and only change the brain from a distance. And they named the experiment that would settle it. Target senescent microglia directly, clear them, and watch whether the wiring holds up. In these mice, aging in the brain's wiring isn't neurons dying. It's the cleanup crew breaking down under its own workload in one exact spot. And that broken state is partly reversible. A support cell, undone by the job it was built to do.